So what you will see about this is then that on the leading strand, or the strand that's furthest from the Y fork, and the strand that's closest to the point of origin of replication, the DNA copy process will happen in this direction, and there will be no problem there. It will be built in one continuous strand with no problem at all. Now on the other strand, because it has to go in a 3 to 5 direction, you're going to have to build on that direction, or towards the point of origin from the Y fork. The problem is DNA polymerase is a huge molecule, and it won't fit on the Y fork. You see, this area here is not wide enough for DNA polymerase to fit. And so here the problem becomes that DNA polymerase cannot copy this piece of the strand because it won't fit there to copy that piece. In other words, it will have to start copying from here, where you see the DNA primase laying down the primer, and where you see the RNA primer. So the DNA polymerase will start from here and kind of build on that direction. Now, what that means is that this piece that was left over here is not going to get copied until the Y fork unzips a little bit more, helicase goes through a little bit more, and now there's more room for DNA polymerase to fit there, which means DNA polymerase is going to have to copy that piece later. So it's kind of like this. If you think about the lagging strand, it'll be built like this, okay? So the leading strand will be built continuously towards the Y fork. So you're going to get the leading strand and build it continuously, the copy, towards the Y fork. Now, on the lagging strand, you're going to build like this. First you build this piece, then you build that piece, then you build that piece. So you're going to make them in small pieces. Each of those small pieces we called an Okazaki fragment, all right, after the scientists discovered it. Then another enzyme kindly comes through to actually make a bridge between the pieces. It's basically what it's going to do is going to stitch up the phosphodiester with phosphodiester bonds, the backbone made of sugar and phosphate groups, and stitch it all up and build a continuous strand the same way it was in the leading strand, which was built continuously in the first place. And ligase is going to be very important to make sure that that's happening. Now, these are the basic enzymes which are going to be involved in the process then. Topoisomerase uncoils and untwists and it stabilizes during the process. Helicase unzips the genes, creates the Y fork. Single binding proteins stabilize the unpaired strands. Primase comes down and lays down the RNA primers, which are the markers for the starting spot of the actual copy process. DNA polymerase comes through as a copy machine and copies the actual code, and it works in 2 strands at once. One machine will copy the leading strand continuously. Another machine will copy the lagging strand in pieces as the Y fork widens and allows itself to connect closer and closer to the actual area that needs to be replicated. And those fragments will then be stitched up by the 6th enzyme called ligase. But you're not yet done. We have more enzymes that need to do their job. And these enzymes are called mismatch repair and excision repair enzymes. Because you see, after this whole thing is done, DNA is not yet ready. By the way, look here again. You see the picture of the process that we described, and you see how the leading strand is built continuously from the origin of replication towards the Y fork. But because the DNA polymerase cannot fit here on the Y fork, the other side is built in segments called Okazaki fragments, and then ligase will have to stitch them up afterwards. Okay? Now, when this whole process is completed, sometimes DNA polymerase makes a mistake. Sometimes it reads the A, and instead of putting the T, it will put a C. Remember that A always needs to pair with a T, and a C always needs to pair with a G. So, you know, let's say you have a sequence, and you're doing like a sequence like A, T, C, G, A, and so forth. That's the strand, that's the template. When I actually build the complementary strand, and I'm going to copy it, which is what DNA polymerase is basically doing, it's going to pick up those triphosphate, nucleoside triphosphates, and you're going to put them in place. Now, over here, it has to put a T, so it goes ahead and put a T. Over here, it has to put an A, so it puts an A. Now, over here, it had to put a C, but by accidentally, it does a mistake, and it puts an A instead. Now, that's wrong. Now, and then it continues going, and maybe it does right from that point on, and it actually does point the C that's supposed to put here, and the T is supposed to put here. But this is a mistake. This is not the right code. And this is what we call a mutation. It's going to be a problem. And it happens because the DNA polymerase is working incredibly fast. This happens at the speed of hundreds of nucleotides per second. And so, ever so often, it will make a mistake and pair up the wrong thing. And that actually is going to make the nucleotide damaged, because, remember, you always bind one ring with 2 rings. And so it would be weird if you paired it up that way, because it'd be like, that's not the way it's supposed to be if you have only 2 rings. Or maybe you even have the 3 rings, but with the wrong pair, because A is supposed to pair with T, or now with C, because T and C are both pyrimidines. So A, if it pairs with C, you would still have a double ring adenine bonded with a single ring cytosine, and you're not going to make the DNA either thicker or slimmer in that area. But that's still a mistake. That still needs to be corrected, because A doesn't pair with C, it pairs with T. And so to fix these problems, you have a group of enzymes which are called mismatch repair enzymes. And all they do is that they identify the area where it's wrong. I call them proofreading enzymes. And they come through and they identify the area that has a mistake. And then a separate enzyme that's called nuclease will do something that's called excision repair. It will actually pick up the actual area that's actually defective and cut it out, as you can see there. Now we have a gap. Now that gap will then be filled in with the action of DNA of primase, which is going to lay down the primers, and then DNA polymerase will come through and build it, and then ligase will come through and stitch the pieces, just like if it was an Okazaki fragment that was added there. This whole process we call DNA proofreading process, and it's involved a mismatch repair enzyme and excision repair enzymes. Now, just so you have an idea of how often this is necessary, the DNA copy process makes about 1 mistake in every 10,000 base pairs. Now, that is really accurate. Can you imagine being that good of a test taker if you only made 1 mistake every 10,000 base pairs? That would be amazing. You get a perfect score almost all the time. Now, if you consider the DNA is billions of nucleotides long, that means that that's pretty bad. Now, 1 in 10,000 doesn't seem that bad, but think about it this way. If you have a house that has a billion screws, if you have 1 in 10,000 screws missing, now you have hundreds of thousands of screws missing. Doesn't it sound that good anymore, does it? And that's the problem. Especially when you consider from the chromosome inheritance lecture that we learned about that a single change can cause a mutation that can make you die. And so it's very important for the body to correct these mistakes. Now, after the excision, proofreading, and mismatch repair enzymes come through and do their job, you're only going to have 1 in every 10 billion mistakes. And now it's pretty much 1 mistake in the entire chromosome, and that's acceptable. And sometimes that mistake happens in a part that's not even important anyways, and we'll talk about that when we talk about protein synthesis. And so there's not going to be a problem. And so life maintains those mutations, by the way. So think about it. Why would you want to keep those mutations? Because think about it. Life could have done better than that. It still leaves 1 in 10 billion mutations left. Well, for a very important reason. If it wasn't for those mutations, you wouldn't have evolution. We would never change. We will always be the same. And since the environment is changing, if we don't change, we will always die, because the environment, eventually we won't be adapted to our environments as a species. And we will always basically be clones of each other. And without the diversity of life protecting us from disease, for example, 1 virus agent that could kill 1 person could kill every person on Earth. Biodiversity makes us more resilient, more likely to survive. It makes life more possible. What are the chances that events will kill all the life forms on Earth? Not that much, as long as you have mutations. And so life has not gotten rid of 100% of the mutations on purpose, because mutations is what actually makes us evolve or change and be diverse. And so they are important for life, so that we can actually adapt to the environment over long periods of time, over many, many generations, as you can see. All right? Now, you could argue, by the way, that we humans are evolving to a different kind of form altogether, as you can see in the picture. All right, but anyways, another thing that's actually important about the DNA copy process is the idea of telomeres. Now, the ends of the chromosomes, or the ends of the replication, actually have a problem. You see, because of the whole Okazaki fragment business, when you get to the end of the lagging strand, the last piece won't get copied. Think about it. You have, let's say you have the end of the Y form, all right, and then the leading strand will not be a problem. You're going to copy that. But what about the lagging strand? You can't copy the last piece, because you don't have anything else to unzip. You're at the end. So the DNA polymerase will never fit there, which means when you finish the copy process, you're going to have something that looks like this. You're going to have a piece missing. That piece won't get copied. And so the problem of this process is that it actually leaves out a piece of the DNA every time the DNA copies itself. So the incomplete ends will be a problem. And so in order to solve this problem, the ends of the molecules actually what we call a telomere. format. It's not actually the double helix that you recognize. It is a special ending to the DNA molecule that we call the telomeres. And basically what the telomeres do is it protects the DNA from degradation, because a special enzyme called telomerase is going to come after the copy process is completed and add the piece that's missing to the end. But that piece is basically junk. It doesn't serve any purpose. It's just a cap that's added to the top of the end of the DNA. And yes, every time you copy, you're going to lose that cap. No copy. But then telomerase knows exactly what to do to add the cap again. And so every time you copy a cell, you lose a cap. And every time, telomerase replaces it. If you didn't have DNA telomerase, that would be a problem. And see, one of the reasons why we age is because of DNA telomerase making mistakes. Because after a long period of time of many copies, and you're always losing more and more pieces of those telomeres, and the DNA telomerase tries to replace the entire cap, sometimes it doesn't. And so after billions and billions of cell copies throughout your life, as you get old, eventually you die because you start losing actual DNA. And cells start aging because they become defective, because they no longer have all the DNA they need because you lost your telomeres. So some people think that our aging process is tied to the destruction of the telomeres at the end of our DNA molecules. And some cancer cells, by the way, do not ever lose the telomeres, which is actually interesting and might be a hint as to why one way to actually reach immortality, by finding out what cancer cells do to preserve them, and then do the same for the rest of the cells of our body. Right? And then maybe we have some hope of not aging the same way we age right now. Although, if you think about it, just like with mutations, there's a reason for aging. A reason why the life made it so that we got older. If we never got older, we will always outcompete the young generation. Our wisdom will always be more than they are. Will always be mismatched, unless of course the environment changed and they had mutations that made the men more likely to survive. But on average, the old generation will always outcompete the new generation, and in that way the evolution will always stop, because the new generation will always go extinct when trying to compete for resources. And so it wouldn't make any sense to even reproduce in the first place. And so aging is a natural process of life in order to make evolution possible. And one of the things that make us age is absolutely the loss of telomeres. There's other things like gathering free radicals over your life, attacking your cells, exposure to radiation causing more mutations, exposure to chemicals and a gathering of chemicals as you go through your life. More and more bad chemicals accumulate in your body, and other things like that. So greater exposure to accidents, which accumulate imperfections in your body. And there's a lot of reasons why we age, and there's chemical reasons for that as well. Some people think that we actually genetically designed at some point to start going old, and some genes are activated to actually initiate the aging process. And so there's many reasons for why we age, but one of the reasons is absolutely tied to DNA copy process, or the loss of telomeres, because telomerase can't always do a perfect job. Thank God for telomerase though, because if it wasn't for telomerase, we would definitely be in trouble, because we would get to the point of death a lot earlier, since we would start losing pieces of DNA every time the cell gets copied. And we would get to the stage we call the senescent cell, when the cell basically kills itself because it has no purpose. It dies of old age, of a natural process. In addition to senescence, the loss of telomeres might serve as a signal that sends the cell into a self-destruct or apoptosis scenario, which prevents the cell from actually developing problems from the loss of telomeres, such as cancer. And so that's another role yet that the telomeres have. And imagine, for example, the offspring. Every time you create an offspring, you would lose a little bit of your DNA. Because when you go through meiosis, you also would have DNA copy happening, and so you also lose pieces. And so every generation, the DNA will get shorter. And so telomeres are very important. So telomerase's job of adding and fixing the end is crucial to maintain us alive. And I did mention that some cancer cells don't have that problem because they fix it, but at the same time, other cancer cells don't have that ability to always correct the telomerase perfectly. So if you think about it, as they reproduce like crazy, the cancer actually ends up destroying itself, because eventually the cells would lose pieces of DNA and die because they're no longer functional because they don't have enough DNA. And so if the cancer does not have the ability to fix the telomeres perfectly, and eventually the cancer cells will actually reproduce so fast, they will lose so much telomeres and so much of the DNA that they will end up self-destroying. So telomeres are actually protective for us in a lot of different ways and very, very important for us to maintain our life.